For our biology project, we are going to conduct 3 different experiments. Our experiments focus on the theme of respiratory system. These experiments are lung capacity, carbon dioxide test using lime water and bromothymol, and lastly, inspiration and expiration test. The aim of this experiment is to confirm a person's lung capacity. Normal lung capacity of a person is roughly 4,500 milliliters. We predict that the results of our team member will be at around the same range. The materials you will need are a water gallon, container, plastic tube, water, marker, and a measuring cup. First, make sure the plastic tube is clean and sterile. Second, fill half of the container with water. Third, mark the water gallon with the accommodated measurements with a difference of 250 milliliters each. 4. Fill the water gallon all the way with water. 5. Put your hand over the top of the gallon to stop water escaping when you turn it upside down. 6. Turn the gallon upside down. Make sure none of the water comes out. Place the top of the gallon under the water in the container before removing your hand. 7. Push one end of the tube into the bottle. 8. Calmly take a big breath in. 9. Breathe out as much air as you can through the tube. 10. Subtract the final volume of water in the bottle from the initial to get the amount of water displaced, which is equal to the amount of air exhaled. Divide this number by 1000 to change it into liters. The result is your lung capacity. From the result, it is apparent that Michael has a lung capacity of 3,000 milliliters. The normal vital capacity for teenagers is roughly 3,500 milliliters. The difference in lung capacity might be because of inaccuracy in conducting the experiment, or the different physical features like height and weight. The aim of this experiment is to determine the fact that we exhale carbon dioxide. Our hypothesis is that after blowing bubbles into each liquid, the lime water will turn cloudy and the bromothymol solution will turn green or blue. The materials you will need are a straw, lime water, blue bromothymol, and beaker. 1. Pour 50 milliliters of lime water and blue bromothymol in 2 separate beakers. 2. For each beaker, blow each liquid using a straw. 3. Observe the results. If lime water turns cloudy, CO2 is present. If the bromothymol solution turns from blue to green or yellow, then CO2 is present. The cloud in the lime water indicates that CO2 is present when we exhale. Likewise, the yellow color in the bromothymol solution indicates that CO2 is present when we exhale. The aim of this experiment is to construct a model that demonstrates humans' inspiration and expiration process. Our hypothesis is that when we pull the balloon at the bottom, the balloon inside will enlarge, indicating the inspiration process. When we release it, the balloon inside will deflate, indicating the expiration process. The materials you will need for this experiment are balloon, plastic bottle, glue gun, tape, straw, cutter, and cork only if necessary. 1. Carefully cut the bottom of the plastic bottle. Put tape all around the bottom, covering the sharp edge. 2. Make a hole on the bottle cap. 3. Insert the tube into the cap of the bottle so that the straw sticks out on both ends. 4. Attach the balloon on the tube using tape. 5. If you can get the mouth of the balloon over the mouth of the bottle, then you will not need the cork. 6. Put the cork into the mouth of the plastic bottle. Make sure it is tight. The sixth step depends on whether you need it or not. 7. Tape the large broken balloon to the bottom of the plastic bottle. Make sure it is airtight. 8. This balloon represents the large muscle that is beneath the lungs, the diaphragm. 9. Push and pull on the balloon slowly to see how it works. If you have trouble catching hold of the balloon, put a small piece of tape on the center of the balloon to make a handle. This model represents our lungs during inspiration and expiration. Pulling the balloons at the bottom of the bottle indicates inspiration. As you can see, this causes the balloons inside the bottle or our lungs to inflate. On the contrary, releasing the balloons at the bottom of the bottle indicates expiration. When this happens, the balloons inside the bottle or our lungs will deflate.